Hey guys, due to some technical difficulties, I've had to repurpose my living room into a recording studio. I guess we're coming to you live from my house and we're going to begin this presentation. Shugoshin, the guardian spirit destroying cancer. Cancer is a group of diseases that has undoubtedly touched many of our lives in some shape, way, or form. Cancer is a very prevalent disease, with an estimated 14 million new cases diagnosed per year, as well as a very deadly one, with an estimated 8 million deaths worldwide per year. And if you look at this graph over here, plotting US mortality rates due to cancer from 2000 to 2014, when we look at this trend line, we realize US mortality rates due to cancer have not really decreased that much in the past 2 decades, which is why it's so important to continue focusing on anti-cancer therapy research. So as we all know, cancer is a result of mutations disrupting proper cell checkpoint function, thus leading to uninhibited cell growth. Another important point is that this misregulated cell cycle will result in certain proteins, often checkpoint associated, to become essential for tumor growth. Targeted therapy is currently a major focus of anti-cancer research, and it uses drugs to interfere with designated target molecules that have been deemed necessary for cancer progression. These drugs work by either directly reducing the activity of the target molecule or by preventing its binding to the receptor. Now, key point here is that these target molecules must be necessary for cancer progression. Now, if we go back to what we just talked about a couple of seconds ago, we see that the misregulated cell cycle will often result in checkpoint-associated proteins to become necessary for cancer progression, which is why target molecules often end up being involved in the cell checkpoint, which brings us to Shugoshin, whose main function is to prevent centromeric cohesin cleavage until proper microtubule kinetochore attachment. So we're going to go and look at exactly what that means with this diagram here of a duplicated chromosome consisting of two sister chromatids just going through mitosis. So here we are in prophase. We have our two sister chromatids that are attached by these cohesin molecules that span the length of the whole chromosome. Now, through the prophase pathway, which occurs in vertebrates, all of the, well, not all, but most of the cohesin along the chromosome arms dissociates, leaving only centromeric cohesin behind. This centromeric cohesin is what maintains cohesion between the sister chromatids during metaphase until proper microtubule kinetochore attachment is achieved, as we see here with the green microtubules attaching to the kinetochore protein, at which point the separase pathway is activated and the centromeric cohesin is cleaved, allowing for full sister chromatid separation in anaphase. Now, the main question here is, how is it that the prophase pathway, which can so easily dissociate cohesin located along the chromosome arms, can't dissociate centromeric cohesin? Now, the reason for that is Shugoshin. So here we have SGO1, which is a type of Shugoshin protein, which localizes specifically to centromeric cohesin. And by doing so, it protects the centromeric cohesin from dissociation by the prophase pathway. And this allows the maintenance of sister chromatid cohesion until proper microtubule kinetochore attachment is achieved, like we just went over, at which point SGO1 will delocalize from the centromere, leaving centromeric cohesin free to be cleaved by the separase pathway, thus allowing for sister chromatid separation. And so by protecting sister chromatid cohesion until proper attachment is made, SGO1 prevents premature sister chromatid separation, which can lead to chromosome missegregation, which brings us to our next function of Shugoshin, which is in preventing generation of chromosome instability, which, if we look at this diagram right over here, is a result of premature sister chromatid separation. Furthermore, it also prevents mitotic catastrophe as well as the rise of aneuploidy and cancer, which result from chromosome instabilities. Now, we'll go more into detail about this later on in the presentation, but for now, we're going to go look first at this first paper, which is a major basis for my proposal. SG01 is a potential therapeutic target for hepatocellular carcinoma. Just want to draw your attention to therapeutic target, which relates back to our previous discussion on targeted therapy. So, some quick background. A previous study found that HeLa cells, which are a cancerous cell line, upon SG01 depletion, committed cell death due to premature chromosome separation. And so the purpose of this study is to look at SG01 and its role in hepatocellular carcinoma tissues as well as normal tissues, and also to look at the essentiality of SG01 for overall cell survival, with the overall goal of the study being identifying SG01 as a potential target for anti-cancer therapy. So, what these researchers first did was they wanted to go look at expression levels of SG01 in both cancerous and non-cancerous cells, which they did using a Western blot, the results of which are shown here. And just by looking at these results, it's fairly easy to see that among the non-cancerous cell lines, which I have circled in red, RPE1 and NEHAP-LXHT, their SG01 expression levels are much lower compared to the other cancerous cell lines. We see this much clearer in this graphical representation, where if you look at these 6 hepatocyte-derived cell lines specifically, we see NEHAP-LXHT, which is the non-cancerous hepatocyte-derived cell line, has much lower SG01 expression compared to all the other cancerous hepatocyte-derived cell lines. And so this suggested to the researchers that hepatoma cells have a much greater need for SG01 compared to normal cells, thus providing further support for investigating SG01 as a possible target in anti-cancer therapy. So, in order to further investigate this, they subjected both the non-cancerous cell lines and cancerous cell lines to SG01 depletion. They did this using siRNA, the targeted SG01 mRNA, in these cells, and they found that the cell viability of the transformed cells following SG01 depletion was significantly reduced compared to the normal cells. And so to further look at this, they observed time-lapse imaging to look at the mitotic progression of these cells subject to either control siRNA or SG01 siRNA. So if you look here at the results, we have HUH7, which is a cancerous cell line, and under control siRNA, the cell simply seems to progress through mitosis, continuing to progress through mitosis until eventually we have 2 daughter cells. I'm not sure how easy it'll be for you to see this, but there's one right there and another right there. So good mitosis. Then under SG01 siRNA, we see the HUH7 cells begin to enter mitosis. However, they appear to be halted in an arrest-like state until eventually apoptosis is induced and the cell dies. Now this stands in stark contrast to NEHAP-LXHT cells, which are non-cancerous, in which both control siRNA and SG01 siRNA, both cells proceed through mitosis, resulting eventually in 2 daughter cells. You see one here and one here, and then in SG01 depletion, you see 1 daughter cell here and 1 daughter cell right there. And so we see how significant this is in cell populations if we look at figure E, where we see under SG01 depletion, approximately 80% of HeLa cells and around 60% of HUH7 cells died in mitosis, compared to looks like about 10% of RPE1 cells and essentially 0% of NEHAP-LXHT cells. Another interesting find was that SG01 depletion induced micronucleation specifically in the cancerous cell lines. So if you look here, this is what normal mitosis looks like with 2 daughter cells. And then here we see micronucleation, where we see 2 little micronuclei right here and right here. This is an enhanced image. And micronuclei actually result from chromosome missegregation, and as a result are a marker for chromosome instability. I'll talk more about this in a bit, but basically the major findings were that SGO1 depletion caused premature chromosome separation in both transformed cells and normal cells. However, transformed cells ended up being more susceptible to cell death. Also, the increase in micronucleation that we just talked about earlier suggests that increased chromosome instability induced by SGO1 depletion could be a possible reason for the selective killing. So now I'm going to go more into detail about that last bullet point. So how can SGO1 depletion seemingly target cancer cells for death? So we're back to this diagram again. So we already know about how Shugoshin prevents chromosome instability by preventing chromosome missegregation. So now if we look here, I've actually labeled these two conditions. So chromosome instability, if significant, can result in mitotic catastrophe. However, if it's just moderate, it can result in misregulation of the cell cycle, leading to cancer. And so it has been proposed that increasing chromosome instability in cancer cells past this apparent moderate threshold could push it to significant levels whereby mitotic catastrophe would result. And with that, we're going to go to the next paper, which is the second basis for my proposal. The intercentromere Shugoshin network prevents chromosome instability. So some background again, this first point should be reviewed at this point. Chromosome instability is a major trait of cancer cells and a driver of tumor progression. Next point, many cells displaying chromosome instability, denoted CIN+, have been found to have an impaired intercentromere Shugoshin network, also known as the ICS network. And so the purpose of this study was to investigate various CIN positive as well as CIN negative cell lines for specific ICS impairment. So this is just a quick look at the ICS network. Just going over some major proteins real quick. So first we have HP1, heterochromatin protein 1, which is important in helping out with the centromeric localization of SGO1. Next we have cohesin, which we've talked about, which is the binding site of SGO1. And then a couple other proteins that I'm not really going to go too much into detail about are CPC, the chromosome passenger complex, which includes Aurora B kinase, a protein you guys have all heard about, as well as BUB1, which is the kinetochore protein, another one you've heard about. This isn't meant to be an exhaustive list, just a quick look at very important proteins in the ICS network. And so what these researchers wanted to do was first observe SGO1 activity in both CIN positive cells and CIN negative cells. And they did this by conducting FRAP experiments using GFP SGO1. So if we look here, we have HCT116, which is a CIN negative cell line, and then we also have 293T, which is a CIN positive cell line. And the researchers conducted FRAP experiments on both. So before the bleach, we see here we have GFP SGO1 fluorescence, as well as we do here. And then after photo bleaching, there is no fluorescence. However, about 60 seconds after photo bleaching, we do see some recovery of this fluorescence right here, signifying that some more GFP SGO1 has diffused into the area and localized there. However, if we look at the CIN positive cell line, we see much more fluorescence recovery. And so these results are quantified here in this plot, where if we look right here, we see HCT116 and HeLa1, which are both CIN negative cell lines, have noticeably longer half recovery times compared to HeLa2 and 293T, which are both CIN positive cell lines. And so the researchers took this shorter recovery time in CIN positive cells to imply that a destabilization of SGO1 localization is what is occurring in these CIN positive cells. And so because building upon that knowledge and the knowledge that SGO1 interacts with HP1 as well as cohesin, they decided to see what would happen upon disrupting these interactions So they did this by generating 3 different mutant forms of SGO1. First we have T346A, in which the cohesin interacting domain of SGO1 is disrupted. Then we have V453E, in which the HP1 interacting domain is disrupted. And then we have this mutant form called TAVE, which is actually a combination of the first 2 mutations, thereby disrupting both the cohesin and the HP1 interacting domains. And they wanted to express these 3 different mutant forms in a CIN negative cell line. And they would observe what occurred through immunostaining, which we see right over here. So if we look at the wild type, we have DNA stained in blue, and then if we look closer in the zoomed in image, the red signifies CNPC, which is a centromere protein, and the green is SGO1. So we see here SGO1 localizes specifically to the centromere. Now if we look at T346A and V453E, we do still see SGO1 localizing to the centromere. However, it's nowhere near as much as we saw in the wild type. And now if we go to the TAVE mutant, we see close to no localization of SGO1 to the centromere. And this phenotype, researchers realized, was actually quite similar to the wild type in CIN positive cell lines. And so these results together indicated that proper functioning of the ICS network, of course, is necessary to stabilize SGO1 centromeric localization. And so any impairment of the ICS network that was seen to be common in the CIN positive cell lines would lead to a lack of SGO1 centromeric localization that could lead to either cell death or further CIN. And so now we get to my proposed study. So, quick review, significant chromosome instability can lead to cell death, whereas moderate chromosome instability leads to tumorigenesis. And as a result, it should be no surprise that many tumor cells display chromosome instability. Now, the first paper we went over basically found that SGO1 depletion can selectively induce apoptosis in tumor cells over normal cells. However, they were unable to determine why. The second paper we went over found that impairment of the ICS network was a common manifestation of chromosome instability in cells, and it was also key in generating further chromosome instability. So in my study, what I propose to do is look at all these 4 points that we have and link them together by investigating the impairment of ICS network as a selecting factor for SGO1 depletion in inducing apoptosis. Now the rationale for this would be normal cells theoretically should display no underlying chromosome instability, whereas most cancer cells do have underlying moderate chromosome instability in the form of ICS network dysfunction. And so from that it follows that SGO1 depletion is, I hypothesize, in cancer cells would exacerbate this impairment of ICS network, thereby increasing the underlying CIN to significant levels, resulting in mitotic catastrophe. Normal cells are able to escape mitotic catastrophe because they are able to withstand the moderate chromosome instability that is generated by SGO1 depletion, as they don't have any underlying moderate chromosome instability already there. So I propose to do this by first determining differences in localization patterns for ICS network associated proteins in tumor cells that are resistant to SGO1 depletion, tumor cells that are sensitive to SGO1 depletion, as well as normal cells. And I propose to do this using fluorescence microscopy coupled with live imaging so that I can track localization patterns of specific ICS network proteins throughout mitotic progression. My next aim would be to assess the differences in chromosome instability in tumor cells that are resistant to SGO1 depletion, as well as in normal cells, both before and after being treated with SGO1 siRNA. And so I would do this using chromosome instability assays that have been developed specifically to quantify CIN in cells. And hopefully the findings from this proposed study would yield further understanding of the role that SGO1 could play in anti-cancer therapy, as well as provide further insight into general mechanism to exploit chromosome instability in order to suppress tumor progression. Thank you. If you have any questions, please feel free to email me, or I guess ask me next time you see me, maybe in class or around campus. So thank you very much, and have a great rest of your day.